grow as the pH changes. However, non-disease-causing staphylococci grow but do not change the medium's color. The combination of mannitol and phenol red allows the differentiation between pathogenic and non-pathogenic staphylococci. Because I was unable to physically do this lab, I'm going to choose Staphylococcus aureus just cause, and the appearance of this bacteria with this technique is round spherical grape-like forms. And this bacteria usually grows in the temperature range of 15 to 45 degrees Celsius. And because I was getting a lot of information that was leading me to think my bacteria was E. coli, I assumed that E. coli grows in 15 to 40 degrees Celsius, which is why I picked this bacteria from the mannitol salt agar lab that we did online. For Magonki agar, it is an example of a medium that is both selective and differential. Biosalts and dicrystal violet make the medium selective by inhibiting the growth of gram-positive bacteria but allowing many gram-negatives to grow. This medium also contains the sugar lactose and the pH indicator of neutral red, which allows for differentiation between non-lactose fermenters. In response to lower pH, neutral red turns from pink to red. The color change distinguishes lactose fermenting coliform bacteria from other gram-negative bacteria. Colonies of lactose fermenters will appear pink. Because non-lactose fermenting gram-negative organisms will utilize peptone in the medium for growth, their colonies will appear clear or uncolored. We were unable to perform this technique as well, but because I know my bacteria is gram-negative, I was able to conclude that my bacteria is Escherichia coli, E. coli. The strong fermentation of lactose with acid from the bacteria causes the growth of bacteria to be bright pink with a halo. And I knew my bacteria was gram-negative from one of the first tests, so I knew that it had to be one of the bacteria that we tested, or organisms that we tested, that was gram-negative. So I assumed it was E. coli because of the bright pink halo, which indicated that there was growth of the bacteria. Okay, for eosin methylene blue agar. It is an example of a medium that is also both selective and differential. The two dyes, which are eosin and methylene blue, make the medium selective by inhibiting the growth of gram-positive bacteria, but allowing many gram-negatives to grow. In addition, this medium contains the sugar lactose, which allows for differentiation between non-lactose fermenters, lactose fermenter, and heavy lactose fermenters. The production of acid while fermenting lactose will trigger a pink to metallic green change that will distinguish coliform bacteria from other gram-negatives and E. coli specifically. Gram-positive bacteria will grow poorly on this medium or not at all. Gram-negative bacteria that are not coliforms will grow well but are basically colorless. Coliform bacteria, which are lactose fermenters, will grow well and the colonies will be pink with purple in the center. Rapid or strong lactose fermenters will grow well and will appear dark purple with a distinct metallic green color. So we were also unable to do this lab, but because my bacteria was gram-negative, I can infer that my bacteria is E. coli. I can assume this because gram-negatives grow very well on EMB, and E. coli was able to grow with a dark purple to metallic green sheen on it based on the colors and the information that I saw online. And that indicated, because of the metallic green sheen on it, this indicated the growth in lactose formation. So it's a strong lactose fermenter. Triple sugar iron agar is a differential medium used for the differentiation and identification of gram-negative bacteria. This medium tests an organism's ability to ferment selected carbohydrates and to produce hydrogen sulfide. This medium contains 3 sugars, pH indicator, 2 sources of sulfur, an iron salt, and a general nutrient base. Organisms that can utilize only glucose will grow and quickly deplete the glucose in the medium. The acids released in the reaction will cause the pH indicator in the medium to change from red to yellow. Once fermentation activities slow down, the pH indicator is the slant portion of the tube will revert to a red color within 24 hours. However, organisms that utilize only glucose will produce a TSIA tube with an acid butt and alkaline slant. The organisms that continue to ferment throughout the incubation period, however, will produce a tube with an acid slant and acid butt, which would be AA. I am going to assume that my bacteria would have been identified as E. coli again with this information because E. coli has a yellow slant and butt color, which makes it an acid, and there was no H2S present, and I conclude that it is a glucose, lactose, or sucrose fermenter. Okay, so for IMVIC, the first test we did was indole. The SIM medium, along with Kovacs reagent, is used to detect indole production. Indole is produced from the enzymatic hydrolysis of amino acid tryptophan. Organisms that produce tryptophanase metabolize tryptophan to produce indole, pyruvic acid, and ammonia. The SIM is inoculated with a needle by stabbing to the bottom of the tube and carefully retracting the needle along the same streak line. Non-modal bacteria will grow in a dense and well-defined area along the stab line, but modal bacteria will swim away from the stab line, and the tube will appear turbid, and the stab line will not be visible or distinct in non-modal bacteria. This is out of order. Usually you do citrate after MRVP, but I just have citrate on the side. Simmons citrate medium contains citrate as the sole source of combined carbon, ammonium salts, and bromthymol blue. Only organisms that import and utilize citrate will grow on this medium. The metabolism of citrate and ammonium increases the pH of the medium, and the bromethyl blue pH indicator changes from green to light blue under alkaline conditions. If there's orange colony growth, this indicates a positive citrate reaction. Before you do the citrate, you do the MRVP, which the methyl red and Voges Proskauer tests are designed to determine if the end products of carbohydrate metabolism are acidic or neutral. A single tube of MRVP broth is inoculated for each organism to be tested. After incubation, the volume in that tube is split into two separate test tubes. The methyl red is added to one of these test tubes, and the methyl red is also a pH indicator that will turn red at pH values less than 4.4. This means that the test is MR positive, which indicates that carbohydrate fermentation produced a stable acid end product. For the second tube, the VP reagents are added, and the reagents react with neutral end products like acetone or butanidol to produce a brownish red color. The tube is VP negative if it remains a yellowish color. From this test, I am going to say that I conclude that my bacteria is E. coli again from the results. I can infer this because the slant color of E. coli is yellow, which earlier I found out that my slant color was yellow, and E. coli is also negative for sulfide, positive for indole, positive for motility, positive for MR, negative for VP, and negative for citrate. The last test that we did, or the last lab that we did, is the antimicrobial susceptibility testing, or the Kirby-Bauer method. The Kirby-Bauer method is a widely used method for testing sensitivity of bacteria to antimicrobials. So, testing if a patient is resistant to certain medicines, for example. The test is done by first covering the surface of a plate with the bacteria being tested. Paper discs containing known concentrations of different antimicrobial compounds are then placed on the surface of the plate. The antimicrobial agent in the disc diffuses out of it, forming a concentration gradient in the agar around the disc. The concentration of the antibiotic at the edge of the disc is high and gradually diminished as the distance from the disc increases to a point where it no longer kills or inhibits the organism, which then grows freely. If the antimicrobial inhibits the growth of the organism, a clear circular area appears around the disc. So we could pick which one of the antibiotics that we wanted our bacteria to be resistant from, and I picked penicillin because I personally am allergic to penicillin. And according to the chart in our lab book, the zone of inhibition, which is in millimeters, is less than or equal to 28 millimeters in order to be resistant to penicillin. So a summary. So all the techniques I was able to do, I for sure know that my bacteria is capsulated, gram-negative, sporadic, spirilla, non-modal, catalase negative, oxidase negative, beta hemolysis. From this information, the first time around, I was completely wrong. I don't know what made me think that my bacteria was this, but I thought that my bacteria was Shigella or Klebsiella based on the information that I had at the time, but I was wrong. And I officially concluded that my bacteria is E. coli, and it was pretty obvious when I looked at the chart because it went completely. I knew my bacteria was gram negative, and then that brings me down here, and then the catalase test, I knew that it was negative, so the only option was E. coli. And that's my presentation.